I think I need to. Okay, so this is the second half. This should actually be shorter. So we'll see. So yeah, I'm going to go through more of the actual anatomy that you'll be looking at. So the nervous system, signaling and neuroplasticity, sensory motor systems, and EEG data, just to look at one example of how this is sort of measured and dissected. And so the nervous system is split into both the central nervous system and like the brain and spinal cord, and the peripheral nervous system like cranial and spinal nerves, which is then branches out to autonomic and somatic. So the somatic branch has voluntary skeletal muscles, so moving, working out, anything essentially voluntary movement related. And autonomic includes heart muscles, smooth muscles, that sort of thing. And so within the autonomic system, there's the sympathetic and parasympathetic system as well. So sympathetic is fight or flight. Fight or flight, so digestion slows down, pupil dilation, heart rate increases, that sort of thing. And parasympathetic is the rest and digest, which is the opposite to that. So this is a pretty simplistic view of neurons, but to be honest, I don't think you need to know that much about it if you're looking more at neurotech equipment. So neurons create an electrical potential gradient, so voltage difference when they activate. So we have dendrites. So this is like the little branches. Let's see if I can find my mouse. So little branches here. So information comes in from those, goes through the cell body, or the little circle, and then comes out through the axon there. And the terminals. And the neurons create an electrical potential gradient when they activate, which draws positive ions towards the inside of the cell. So you can just see that with the positive ions here moving into the neuron. But the communication between neurons happens at the synapses. Let's see here. Oh, looks like it's not gonna, it's not gonna show the video. But basically, for the synapses themselves, actually, I think we're just going to have an empty slide, unfortunately. So when the action potential reaches the synaptic terminal, it causes sodium channels to open. Those sodium ions release into the neuron, triggering the release of a neurotransmitter. And this then causes the release of neurotransmitters into the synaptic cleft and binds to the next neuron. And so that's just, you're just moving from one neuron to the next. And, it is basically just has to do with the signal in between neurons. But again, that's getting pretty into the weeds of what that would look like. So important pathway to keep in mind is the sensory motor pathway. And this is also, I think, a pretty applicable one usually. So we have sensory receptors, which transmit a signal or impulse to the brain. So you might have the experience of just feeling cold water, for example. And so you have the sensory endings in the skin. And then this goes up along nerves to the spinal cord and into the brain. It reaches the thalamus, so it goes up here, all the way up to the thalamus here, and which relays sensory information from your body to your brain and then reaches the sensory cortex. So if someone were then to decide to initiate movement, like move their hand away from the water, maybe, it's too cold, maybe you want to change the temperature of it, the motor cortex then generates an impulse, which generates that voluntary movement. And this impulse then moves out of the brain into the spinal cord, down the arm, and through the neuroskeletal junction, resulting in the movement of the hand. So you can see the red here, all the way back down, and then back into the hand there. But this is different from pain. So whereas that would be a voluntary movement, You can also get reflexes that would just kind of happen in the spinal cord alone. So this pathway is if there's, so this whole pathway is if there's a conscious choice or decision in that movement, but there's also a reflex that's only communicated in the spinal cord. If we were to move our hand away, as like if you have pain and you suddenly move it away, and then you have the conscious realization afterwards that something was too hot, that comes from that reflex. So we can look then at how they communicate, at the signaling and neuroplasticity. So as I mentioned before, when we're measuring biosignals and neural activity, we're measuring the action potential. And I mostly just kept this slide because I really like the voltmeters that are attached here. But this is just to remind me and you that there's electrical activity that we're measuring. But unfortunately, we can't actually just get straight into there and measure each neuron like that. So instead, we look at the signal that they're giving us here. So as I discussed with the synapse, the neuron receives stimulus, like from that of the environment through a receptor or a signal from another cell like neurotransmitters. If the stimulus is sufficient, it triggers an action potential. So you can see the threshold, you can see the stimulus here. It reaches depolarization, reaches kind of a climax, repolarization, and then a refractory period here. And so this is what you're generally, as one of the examples, what you're looking for when you're looking at EEG readouts, is you're looking at this. So yeah, the neurotransmitter can either help and excite or hinder or inhibit neurons from firing this action potential. And they're all or nothing here. And then this is another image that I already showed, which showed the temporal and spatial resolution of this neural stimulation or neural imaging. Again, showing kind of electrophysiology here, and then kind of going outwards, you can see the fMRI like I illustrated before. And so yeah, electrophysiology is high on temporal, low on spatial. So it's better for time series data or looking at when things occur, but the electrodes don't give specific locations, like I said, or you can only get it based off of where the electrode is. You can kind of do this a bit more with kind of invasive single cell electrodes inside the skull, but it's again not done in humans. And then there's metabolic, which is low temporal, high spatial. And so we also have neuroplasticity, where cells that fire together wire together. And in humans, we record groups of cells. And so cells that tend to communicate together will exert more energy to communicate with those cells. And when they don't communicate together, there isn't as many connections. And so we've mainly been talking about what happens with neurons, what sort of signal it is, what it looks like, but we should also look at where that might be and isolate where it's coming from to help you sort of understand what's going on. And so if we were to look at sensory motor systems, we have our sensory system in which we perceive the world through that environmental processing, and it results in some sort of action decision and then motor output. So again, this is what I was describing with the sensory motor system before. And then we can act or move based off of that information, and it becomes a feedback loop or that monitoring. And so there's a few sensory systems to consider: auditory, visual, and tactile. Auditory is in the temporal lobe in the A1 area, which is right by your ear, which makes sense, right here. The visual system is the occipital lobe, V1 to V5. So that's the back of the brain here. And so you have a couple of different pathways, both of like what you're seeing and where you're seeing it. And then you have the tactile or somatosensory system, which is in the parietal lobe, area 1 and 2, and my favorite, which is the homunculus here. You might have, so we have like a different number of neurons that are associated with tactile sensation, um, which is illustrated by this guy, this homunculus. Um, so uh, it's showing kind of like the ratio or like how many neurons we have or how many, how we're perceiving things in the body. So hands take up a lot of the sensation as well as the lips in humans. Um, and uh, what I find kind of interesting is that I think the homunculus has been like kind of measured and mapped out for quite a while, but female attributes of the homunculus have only been mapped out probably in the last decade, something like that, and they're just doing a lit review on it now. Um, So also kind of in sensory systems altogether, something that I was particularly interested in was the intersection of them, so multisensory integration and how you kind of navigate the world through sensory systems. And like how much crosstalk there is. So often you're not experiencing something through just one sensation, you're experiencing it through multiples. And so one example that was actually part of my master's was peripersonal space, which is essentially the action space around your body. It's how you navigate the world. It's where you know where your body is in space and how you can like use equipment and tools around you. And that requires all three of these. It requires auditory, visual, and tactile stimulation, information together that's in combination and it's integrated all together. Because if you were to understand the space around you, you might hear something farther away, you might hear rustling next to you, and it's all going to help with how far away something is. Visually, we have that same thing. We have the 3D sort of experience around us, and then we have tactile. We have what's physically kind of in front of us, what we are like sensing and feeling all at the same time. And so these are all kind of combining together for us to be able to navigate the world. So things are generally a little bit more holistic, essentially. And then this communicates with motor systems too. So we have cortices like the primary motor cortex, and this is involved in things like proprioception, which is where your body is in space. Like, I know I just said that, but it's like it's the feeling of your body and like the kind of like movement of where your body is, the muscle kind of contractions. Like if you were to close your eyes, you'd still kind of know where you are. And that would be part of proprioception. So again, it has more to do with the feeling of sort of your body um, in the environment. And then premotor and supplementary motor cortices have to do with planning and action of movement. And so this action planning makes more sense with this context, but you can also see it in a slightly different way. And this is part of where my research or kind of my background comes in again, which you can actually see it in a slightly different way as well, which is that we have sensory coding that results in the perception and action planning, and then the motor programming that happens. So again, you have sensing something, deciding to do something, and then doing that thing. But you also have parallel processes that are happening without that decision making. So you also have times in which you are sensing something and then acting without anything kind of going on in between. So one example of that is, again, like reflexes, ingrained actions, sort of habits, loops, fast loops of things that are going on at any given point. But there's also like research kind of in embodiment about how decision making isn't something that is necessarily something that you consciously think of, but you might actually decide through your body to do something like grab a bag of chips before you've decided consciously that you're going to grab the bag of chips. And so all that to say that it can be slightly more complicated in terms of what that would look like. And so we can look at a bit of what all of this kind of looks like with EEG data, which includes brain waves. So these are oscillations or like rhythmic changes that happen as a group of neurons are synchronously firing. So basically there's a rippling or resonance and we get this brain wave. there's different frequency bands that are associated with different types of activity. So you can see that on the right-hand side here. So delta is associated with deep sleep, theta with meditation and creativity, alpha with relaxed reflection, beta with problem solving, and gamma with heightened awareness. And some of these are easier to detect in certain areas of the brain. So, for example, alpha is, I think, one that's associated with sort of visual processing. Yeah, the visual cortex. But there's also another one, which is mu. It's not described as often, but it's, I think, kind of described as a subset of alpha, and it definitely overlaps, but it's found over the motor cortex. And that one's interesting because that one potentially has to do with empathy. It has to do with motor neurons and your sort of perception of someone else, and your action of someone, or like the way that you perceive someone else's actions in the motor cortex. And so when we measure with an EEG, there's different ways we can slice it. So this is all kind of with the same data. We can just slice it in different ways in order to look at sort of different things. And so this includes frequency, which is what I was just talking about. It's time, it's space, and then it's time frequency here. And so you can see how these are sort of split up. So I think that with frequency, can also be used with the Muse headband. So it's marketed to like observe your own brain waves. And this one I think just has to do with like, yeah, so it's just like, I think it just has to do with like which one is there's more of, essentially. Times slice is one that's really common in a lot of cognitive tasks. So frequency is used too, they'll often report it, but time is used because you can chunk time. So you can see here that this was probably in milliseconds, and it's sort of what's going on in a time series. And so you can look at triggers, you can like you can record when a change in a screen happened, like what someone's looking at, and then you can record how they responded to that change, and essentially takes time slices from that. And that's where you get like the little waves. And then you can also like locate it as well. But EEG is also very noisy. It picks up on a lot of different information. So because we're measuring these biosignals and these impulses, we're also getting them from other areas too. So if you run participants in the lab, they'll often sit down with a cap, and you'll have them like blink, like just exaggerated blinks, or they'll like rub their jaw or like kind of gnash their teeth. And you can actually watch as that will create more noise on the computer. And that's mostly to show participants that the more they move, the messier the data and the more difficult it is for us to sort of understand. But it's also just like another example of the fact that like you're reading electrical impulses from like everything, right? So you want to try and isolate it to the skull, but you're going to start getting it kind of in different parts of the body if you're moving in different parts of the body. And so this includes like blinking, breathing, heartbeat, electrical noise, muscle movements, sweat, tongue movement, head or cable movements. So if you're actually accidentally moving the amplifier. And so you can do some of these things like build the chambers, like the Faraday cage, like I showed, to reduce some of that environmental noise, or you can use LED lights instead, which can help over like incandescent or fluorescent lights, because those can also contribute to that. And so we can extract eye blinks as well. We have the raw and processed EEG here, and then we have the extracted eye blinks. But it's a balance between signal and noise, and it depends on just how much data you have and on the equipment that you have, how much you can extract of all sorts. And so there's a few EEG limitations to consider. So this includes poor spatial resolution. So the less sensors you have for something, the less you can map it to location, like I said. So if you have 12 electrodes, you can get kind of like a coarse level understanding of where that is on the scalp. And something with research that has 256, you're going to get a much fine-grained detail. But even then, you're just getting sort of the impulses around the scalp. You're not getting anything inside, right? Not like an MRI. And then there's also artifacts, like I just mentioned. There's also how alert someone is, if they're hungry, if they're on certain medications. I actually did the Muse once for like an open house, and I was told that I had the noisiest brain waves of everybody there, so I take that trophy home, probably because I have ADHD. And so often they just won't even run people with ADHD because it's just pain in the butt to try and deal with the noise of that data. And so locations that are deep or small are difficult to capture, both because of the EEG itself and again, the number of electrodes that you're using. And there's just some tasks that it's not suited for. So if you're looking to locate something or it takes a long time, it might not be captured very well. And so something like fMRI is better suited for that. But not very many of us have an MRI that we can use. And so that is everything for that workshop as well. Yeah. Is there any questions for this one? If not, thank you. Thank you. If not, I think I reduced it in half. We're early night. Okay. Cool. Well, you can also just come up and if you have any questions too, if you think of anything. And yeah. Yeah, thank you for coming.